It's a great pleasure today to introduce to you Professor Sir David Klenerman, that has been our keynote speaker in the PhD and Postdoc Symposium this year, 2025. He's a professor in biophysical chemistry at the University of Cambridge, co-inventor of the next generation sequencing technology, and co-founder of the Solexa company, now known as Illumina.
So the Human Genome Project was finalized in 2003. It was a huge collaborative effort that took approximately 10 years between different countries, institutions, and companies. Professor Klenerman, so you just finished your postdoc at Stanford and went back to Cambridge, and together with Shankar Balasubramanian, you developed the sequencing by synthesis technology. With single molecule fluorescence, Shankar and you managed to read the 4 DNA bases in a massively parallel fashion that was is what allowed now to sequence human genomes at a rate and cost that before was unimaginable. No? So did you imagine, or were you aware at that time of the implications and impact that this technology was going to have over the next years?
So no, I mean, I think we both just moved to Cambridge and started our research groups. And like I described in the talk, we were actually trying to do a much more fundamental basic experiment, which is watch a DNA polymerase copying DNA. And while we were doing that, we realized we could just tweak our experiment to come up with a way of doing DNA sequencing. And then we did a sort of back of the envelope calculation that suggested it could be very fast. And so we just thought, well, this is a very exciting idea. We don't know if it's going to work or not, but we would like to try out the idea. So our main focus was getting into a position that we could actually try the idea. And we decided that we'll form a spin-out company because we were convinced that we need increasing amounts of money in order to develop the technology and develop it fast. And that turned out to be the case. And originally we just imagined that it would be a discovery tool for scientists. So the way it's taken off across the world was well beyond our expectations. And I just wanted to correct one thing. We came up with the idea, but it was a large number of talented people from a whole variety of different disciplines that took the idea and got it to work and then improved it so the cost is, so it's so fast and so low cost that it is today.
Yeah, it continued improving through the years. Yeah. Thank you.
And so looking back, what discoveries or implications in the sequences by synthesis technology that you developed has surprised or excited you the most, especially in terms of implication in medicine or society? And if I may add another part, how did this important development influence your perspective about your work and your research?
So I mean, we originally, like I said, imagined it as a discovery tool because at the time it wasn't clear how much or how little your DNA influenced your predisposition to get diseases. So the speed that it was taken up and used clinically, I think surprised us, both of us. So as you know, it's used to diagnose cancer and for some sort of precision medicine. It's used prenatally. You can take the blood of a mother, and there's a small amount of the DNA from the baby in that blood, so you can do prenatal screening without having to do any interventions on the baby. And you can look at circulating DNA for cancer. And certainly in the UK, like I said in the talk, at Adam Brooks Hospital, they were sequencing children with rare diseases, and they could work out in about half the cases what's wrong with them. So the speed of take-up medically has been, it's very rewarding to see it, and it was really unexpected that it would happen so fast. So what was the second part of your question?
How did this important development influence your perspective about your research?
I mean, I think what it did was give, I think, both of us confidence that our ideas if we had ideas that sounded exciting, they might actually work and be exciting. So it gave us a lot more confidence to try out other ideas, some of which have worked quite well, but nothing as well as the Selexa idea, and some which haven't worked. But I think it gives you confidence that you can actually work well as a scientist, that you can generate new ideas, and some of these might have some impact. So it just empowers you to come up with ideas, persuade my students and postdocs to try them out, and sometimes, you know, we can discover other things that lead us in different directions. So I think it was just a big confidence booster ultimately in our abilities to be scientists.
And also, what type of limitations did you encounter during the development of the methodology?
I think, I mean, the biggest single thing was to persuade the VC to give us money to get started. And it was problematic for the VC because normally people come to them with some proof of concept that the idea will work. And generally the concept is something that's maybe a tenfold improvement in the technology, or enabling one to do something better than other technologies. But what we came to was entirely the opposite. We came to them literally with an idea written down on a piece of paper. We'd done no experiments. Then on the other hand, it was clear that if we could get this to work, there was massive impact, because everyone was saying they wanted to sequence human genomes for less than $1,000. So they didn't quite know what to do with us. And so they spent about 6 or 9 months debating whether they should fund us or not. And eventually, fortunately, they decided to fund us, initially at quite modest levels, a couple of postdocs to get started. So that was probably the biggest problem. I've been involved in other spin-out companies, and I think a strength of what we were trying to do was essentially we just had to do one thing and get it to work. There was we did debate, you know, if we could sequence 1 or 2 bases, could we do with it? But we always just came back to sequencing at least 25 bases and really doing what we set out to do. So it wasn't really any opportunity to deviate from the plan. And then, you know, there were various technical hurdles that the team of people working on systematically solved. And once we could do one cycle, the question was, can we do another cycle and can we do it better until we could do 20 cycles? So I think it was getting started was really the biggest problem. And then, you know, we had no experience of being involved in a spin-out company, and so we had to learn a lot about patents. We had to learn something about business. The VC was very good at bringing people in to complement what we could do. But I think it was just initially getting the whole thing started that was the biggest issue. And then it probably, there were still issues, but they were less than the first issue.
Thanks.
And what do you think is the future direction for single molecule fluorescence, and if you have inspiring new projects, or how you are focusing your research now on?
I mean, we continue to work in the single molecule fluorescence field around 3 main problems. The one we have the biggest effort on now is trying to understand the molecular basis of neurodegenerative diseases, where a monomer binds to another monomer to form a dimer, and then you get protein aggregation ending up as these plaques and tangles that you see in Alzheimer's disease brains. And it's these much smaller aggregates that have been essentially invisible to most techniques that are supposed to be thought to be the damaging species. And so we can actually visualize these species, and we can visualize them in human samples. So we have the opportunity to start to understand how these aggregates form and how they spread through the human brain. And as a result of that, we can actually see these aggregates in blood or saliva, and they change as people develop the disease. So we're very excited that we might have used these methods for early diagnosis of neurodegenerative disease, which I think would mean that any treatment that you had would be much more effective if given early or pre-symptomatic. The other things we're working on are more basic, and essentially how the adaptive or innate immune system recognises a pathogen or a cancer cell and how it decides to initiate an immune response. And again, both systems are very sensitive. They can detect one or a few pathogens or a non-self peptide and a seal self peptide. So we're trying to understand the molecular mechanisms that underpin those processes. And the innate immune system is linked to the neurodegenerative disease because we think inflammation is a major driver of the disease. So we've moved, much more from what was a relatively simple problem in retrospect, DNA polymerase copying DNA, to a much higher level of complexity, the cellular immune response, and also neurodegenerative disease in humans, which is very complicated. So we've really applied what we've been doing to more and more difficult problems, with the view that we'll go for the most interesting and important problem. In the worst case, we'll fail and we'll retreat back to do something a bit easier.
Yeah, I think it's a very interesting example of how with a technology or a set of them you can find every time different applications, and improvements that will be important advances in the biology field, in the medicine field.
I mean, the important thing to note is we've tried lots of other things and they didn't work, so I don't talk about them. So I'm talking about the things that did work. So some of these problems, we realised when we got into them, we were very well suited to our methods. There are other problems that we were very interested in which weren't that well suited to our methods, or our methods simply didn't work. So we've done a lot of experimentation to find these problems that we're working on.